Hello, everyone, and a very warm welcome to Kognito Key. I am absolutely thrilled to have you here today. In today's explainer, we're going to explore one of the most fascinating and, quite frankly, vital laboratory techniques in microbiology. It's called the hanging drop method. If you've ever wondered how scientists manage to look at tiny, living creatures without harming them, well, you're in exactly the right place. So, here's the big question. How do you actually observe living, moving bacteria without destroying them? You see, in a standard microbiology lab, we often use a process called heat fixing. We do this to attach bacteria to a glass slide so we can stain them and see them clearly. But here's the major challenge with that. Heat fixing literally kills and destroys the bacteria instantly. If we want to study life in actual motion, to see how these microbes truly behave and swim around in their environment, we simply can't use heat. We need a totally different approach. Okay, let's dive right into this. The brilliant answer to our challenge is the hanging drop method. This is a highly specialized wet mount technique. Instead of squishing the bacteria flat under a heavy piece of glass, we actually suspend the living microorganisms in a tiny drop of fluid, usually taking it straight from a liquid broth culture, and we let it hang over a concave glass slide. This is amazing because it gives the bacteria their own little three-dimensional swimming pool to move around in. It allows us to see them in their beautiful natural state, keeping their true shape and their motility completely intact. And you know what's truly fascinating? This brilliantly simple yet highly effective technique isn't some brand new technology. It actually dates back to a single powerful year, 1878. It was first discovered and utilized by the legendary scientist Robert Koch. He desperately needed a way to carefully observe living cells, and this elegant solution he came up with has remained an absolute staple in microbiology labs for well over a century. All right, moving on to the lab setup. Let's talk about preparing the drop. Here's exactly how we prepare it, and honestly, the genius is all in the simplicity. Step 1, we take a clean, very fragile glass cover slip and dab just a tiny bit of petroleum jelly on all four corners. This petroleum jelly is absolutely crucial here. It acts as a perfect seal to prevent our liquid from evaporating, and it blocks out any disruptive air currents. Step 2, we carefully place a single loopful of our microbial liquid suspension right in the dead center. Step 3, we take a special cavity slide, which has a small bowl-like depression in the middle. We turn it upside down and gently press it right onto those jelly corners. Finally, step 4, with a quick, smooth motion, we invert the whole thing. And just like that, our little liquid drop is literally hanging suspended inside that glass cavity. Okay, once our slide is perfectly prepared, we move over to the microscope phase. We place our slide carefully on the light microscope. Now, I have a brilliant little trick for you here. Because the drop is clear, finding it is actually pretty tricky. So we start with the 10x objective lens and we scan around looking for a thick, dark, curved line. That line is the very edge of the fluid drop, which happens to be exactly where the bacteria love to gather. Once we find that edge and get it in focus, we switch over to our 40x objective lens for a beautifully clear, high-resolution view of the microbes swimming around. And of course, because these microbes are fully alive, step 4 is essential. We must carefully dispose of the slide by soaking it in a strong Lysol solution for at least 15 minutes to safely decontaminate it. So now that we're looking through the microscope, what are we actually seeing? We have to be very careful here to distinguish between two things, true motility and Brownian motion. Brownian motion is basically a visual trick. It's just the erratic, random bouncing of bacterial cells caused by water molecules constantly bombarding them. They're just jiggling in place. True motility, on the other hand, is actual self-propulsion. This is when we see bacteria actively swimming, gliding, or using their little tail-like flagella to move deliberately across longer distances, zipping around in multiple directions. And this brilliantly illustrates exactly why the hanging drop method is so essential for us. Some bacteria, like those spiral-shaped spirochetes, are so incredibly delicate that standard heat fixing destroys their shape completely. This hanging drop method lets us study them without a single bit of distortion. Furthermore, because the cells are alive and happy in that little drop, we can actually sit there and watch live cytological changes. We're talking about cell division or spore germination happening right before our eyes in real time. We can even clearly identify tiny internal structures like vacuoles and granules. It's like having a front row seat to microbiology. Of course, every scientific technique has its balance. Let's look at the pros and cons logically. The major pros are pretty clear. It beautifully maintains the cell shape and its living state. Plus, that clever petroleum jelly seal means the drop won't dry out, which allows us to observe the slide for a very, very long time. However, the cons bring some very real lab risks. First off, it is highly risky if you are studying live, dangerous pathogenic bacteria, precisely because they are not killed during preparation. Second, it's a very delicate process. Those cover slips break so easily. And if you use too much petroleum jelly, it can squeeze right into your sample, completely ruining your results. Or even worse, sticking to your expensive microscope lens. To wrap things up, I want to leave you with this defining thought from our source material. The hanging drop method is the best way to study the different events of the organism in a living state. It truly is a remarkable window into a hidden, microscopic universe. It lets us witness the uninterrupted dance of life exactly as it happens in nature. It really makes you wonder, doesn't it? What other invisible worlds are waiting right under our noses, just needing the right technique to be seen? Thank you so much for joining me on this microscopic journey today. If you found this explainer helpful and fascinating, please hit that subscribe button for Kognito Key. Make sure to like and share this explainer so you can support us in creating more deeply detailed science content just for you. Keep learning, keep exploring, and I'll see you in the next one.